Hello, my name is Anna Urena, and this presentation is over Herceptin treatment for cancer cells. So a regulatory functioning cell cycle allows cells to grow, replicate, and divide, in which this is all controlled by complex series of signaling pathways for the DNA replication to take place and divide. For cells to function properly, they have regulatory mechanisms such as contact inhibition, which is a process of arresting cell growth when cells come into contact with one another. These forms of mechanisms are vital for the cell since they prevent errors and corrects them when present. However, if errors in cells are not corrected, the cells recognize this damage within them and will commit suicide or self-destruct, which is a mechanism known as apoptosis. So the cell cycle undergoes G1, then it goes through the S phase, then it goes through G2, and then it goes through mitosis. And this is the regular cell cycle of a functioning cell. As a result of genetic mutations in cancer cells, there is a malfunction in this regulatory process, causing uncontrolled cell proliferation in the body. For the case of breast cancer, it involves cells that overexpress the HER2 gene. Therefore, they're said to be HER2 positive. Herceptin's chemical name is trastuzumab, and it is used to treat HER2 positive or human epidermal breast cancer receptor 2 cancer cells in early or advanced metastatic stage to stop the cancer from growing. So Herceptin is a part of a drug class called biological therapies, which are monoclonal antibodies and fusion proteins that incorporate specific recognition components with antibody function. This HER2 targeted therapy is humanized antibody that binds to the product of HER2 oncogene. It works by attaching itself to the HER2 receptors on the surface of breast cancer cells and blocking them from receiving growth signals. Patients that have metastatic stomach cancer may also receive treatment of Herceptin. Now in figure 1, you can see a visual representation of breast cancers having an HER2 protein on their surface responsible for sending signals to the nucleus to divide. And then within this, you can also see an overexpression of HER2 receptors in an abnormal HER2 positive breast cancer cell that it displays this overexpression and a dysregulation receptor. And then you can also have see figure 2, how Herceptin works. So it kind of gives you a little visual representation of how it blocks the receptors and stops the signal responsible for cancer cell growth and division. In figure 3, it is a mechanism of action of Herceptin and how it's used in clinical practice. One can see that the extracellular domain of HER2 can adopt a fixed conformation resembling a ligand activated state, which permits it to dimerize in absence of a ligand. Now, the receptor overexpression can include dimerization. Once activated, the signal transduction cascades from these receptors, promotes cell proliferation and survival. The cleavage of the extracellular domain of HER2 leaves a small remaining at the cell membrane, which this is basically what can be visually seen in this figure. So the side effects of Herceptin, although Herceptin is an HER2 targeted therapy treatment that targets HER2 positive cancer cells, it may also affect normal cells. Herceptin is not for everyone, which is why a doctor should be consulted and tests should be run before starting treatment, especially those patients that have heart conditions, severe lung problems, low white blood cell count, and or pregnancy. And here is a list of side effects that patients have reported while receiving this treatment. So patients that have breast cancer and patients with stomach cancer. So the history and discovery of Herceptin. The promise of Herceptin actually began in 1979 when a pioneering cancer researcher named Robert Weinberg identified the HER2 gene that is involved in multiple cancer pathways. Later, GeneTech was actually the fledgling biotechnology company in South San Francisco that brought Herceptin to the market. These were the early days of Herceptin before it was actually approved. In 1984, a GeneTech scientist named Alex Ulrich identified the protein produced by the HER2 gene. Later in 1986, Dennis Slamon and Ulrich hypothesized together that the HER2 new played a role in growth of breast and ovarian cancer. So they actually researched together. In 1987, they published results suggesting that cancers overexpressing HER2 are more likely to reoccur and spread quicker. Then Herceptin entered clinical development in 1992 and was approved in 1998, initially for the treatment of HER2 positive metastatic breast cancer. Later in 2006, the use of Herceptin was expanded to include post-surgical treatment of HER2 breast cancer based on a 50% decrease in risk of relapse and a 33% decrease in risk of death. So figure 4, you can see the chemical structure of Herceptin. And in figure 5 is an image of the researcher Robert Weinberg, who's an MIT research professor. And questions is, how are patients monitored when receiving treatment for Herceptin? And then question number 2 is, what is the mechanism of action for Herceptin? And this figure 6 just kind of shows you a little visual representation of the identification of HER2 amplification. Here are my references, and thank you so much for listening.